So I think next up is Jeff Rubens. Jeff, please come on up.
All right, great. Well, thank you for having me out here today. It's always a pleasure to be able to share my research with all of you. You saw from Taylor's talk, which was excellent, that he was the 2020 NextGen award winner from CCF. I go back a few more years. I was the 2017 NextGen award winner, and I think you'll hear much more about this right after me, but you know, that funding is so critical. Oh, am I going without me, huh? It's so critical for my early career development to sort of set up my lab, to transition to faculty at Johns Hopkins, and you know, I thank you for that early investment in my career. So, to get started, so you know, in my lab, what we do is try to figure out new therapies that are going to help improve treatments for childhood brain tumors. Oh, keep going without me. Sorry about that. One of the ways that we do it is that we try to figure out what's different about the brain tumors compared to normal cells, so that we could target these abnormalities, improve treatments. One of the tumors that we focus on is the atypical teratoid rhabdoid tumors that you could see here. This is the most common malignant brain tumor of infancy. It's treated with very aggressive treatments, multimodality therapies that include surgery, intensive chemotherapy, autologous bone marrow transplants, radiation, which invariably disrupt the children's development and lives. But all too often, it's not enough therapy to cure these diseases. So, you know, for all these kids, but especially for the kids who have progressive or relapsed diseases, we're really in dire need of developing new therapies. So to figure out how to target these tumors, we have to sort of go back to what drives the tumors. So in a lot of adult tumors, you have an accumulation of genetic mutations over a lifetime that causes the tumors to grow quickly or resist cell death. In childhood tumors, you often have mutations, relatively few mutations, but just in the wrong place. And so in tumors like ATRT, you often get mutations in something that causes a disruption of sort of the machinery that controls the regulation of gene expression or epigenetic modifiers. In ATRT, we see a deletion in the SMARCB1 gene that disrupts the SY-SNF chromatin remodeling complex. And one of the things that does is it increases the activity or expression of this EZH2 methyltransferase, which uses a methyl group that's donated from S-adenosyl methionine, or SAM here, that we'll talk more about later. And it uses that methyl group to methylate its histone target, H3K27. And one of the consequences of increased H3K27 methylation is that you maintain these tumors in sort of an undifferentiated state, in an embryonic stem cell-like state, that are highly resistant to therapies, that you know, grow rapidly and lead to these poor survivals. ATRT, as Jean had alluded to earlier, are more complex. So a lot of these brain tumors have many molecular subgroups that all are different entities. So in ATRT, there's 3 molecular subgroups. They all have different activated pathways that sort of drive their growth and survival. And they all have distinct responses to therapy. So it's important to understand the differences between these molecular subgroups in order to develop targeted therapies. In our project, we focus on the mixed subgroup of tumors, both because there's some data that shows that it's especially aggressive and difficult to treat, but also it's an abnormality that's shared with a lot of aggressive tumors, a lot of aggressive brain tumors like medioblastoma, other tumors too, like neuroblastoma. And so we look to see, what does MYC do to these tumors? And one of the things that we know that MYC does is that it helps reprogram how these tumors generate their energy to support this rapid growth. And so for our tumors, we took our ATRT cell models. We have 8 of them. We did a Western blot just to see which ones express MYC. We see that half of them express high levels of MYC, half of them are very low levels of MYC. And we extracted metabolites. We ran the metabolites through this high-performance liquid chromatographer mass spec, which separates out the metabolites so that we could figure out how just the differences between the metabolic profiles between the high-MYC and low-MYC cell models. What we see is that the high-MYC cell models have a unique metabolic profile. They separate out compared to the low-MYC cell models that you could see in green. And when we look more specifically at the specific metabolites that are responsible for these changes in metabolic profiles between these tumors, we find that two of them that stick out are glutamine and glutamate, so two metabolites involved in glutamine metabolism. And in specific, when you look at the ratio of glutamine to glutamate, we find that the high MYC expressing cell models have an increased ratio of glutamine to glutamate. And what this meant to us before we did anything more was just that somehow these high MYC expressing cell models were using glutamine in a different way than the low MYC expressing cell models. And so we took our different cell models and we titrated glutamine into the media to just look at what is the relative dependency in the high MYC expressing cell models versus the low MYC expressing cell models. And we find that the low MYC models in green there grow relatively normal and very little glutamine. It needs about 0.5 millimolars of glutamine to grow fairly normal. High MYC cell models, on the other hand, really are dependent on glutamine for their growth and survival. So with zero glutamine, you can see the representative pictures in culture, the cells just don't grow. And it takes nearly normal levels of glutamine to support the normal growth of these high MYC lines. And so taking that information, we decided to try to target glutamine metabolism in these high MYC ATRT. And so we used a medication called DON, or 6-diazo-5-oxo-L-norleucine. This is a medication that's been used in pediatric clinical trials before. It's been shown to be safe in clinical trials, but it's never been specifically used in MYC-driven cancers. So we took it in our ATRT cell models, and I just show the high MYC cell models here, but high MYC cell models were very sensitive to DON treatment. So 15 micromolars of treatment really wipes out growth, as you could see in the representative pictures of culture after 5 days. You see the MTS assays below, which show that at these relatively low concentrations, you're decreasing cell growth. The low MYC models I don't show here require much higher concentrations, 50 or 60 micromolars to have the same effect on cell growth. DON also induced apoptosis, which I don't show here, but when you develop orthotopic mouse models of the high MYC expressing tumors, we treated them with just weekly DON. It was tolerated very well by the mice, and you could see it nearly doubled survival in this high MYC expressing model. This exciting results. We're thrilled about it. But the disappointing part is that these tumors continue to grow. The mice eventually die from further tumor growth. And so we wanted to figure out what exactly DON was doing to these tumors so that we can figure out sort of a rational combination therapy to enhance the survival effect. And so a talented postdoc in my lab, Satoshi Nakata here, went back to the initial metabolomics data to see what DON was doing after you treated these tumors. And so one of the things that he found that sort of stuck out was that this ratio of SAM to SA was decreased significantly after DON treatment. We thought maybe this was a result of just cell death, so we treated these cells with lethal doses of carboplatin, and you see no effect on the SAM to SA ratio. And so if you remember from the beginning, why that's important is that SAM is this universal methyl donor. So you get increased activation of EZH2 in these ATRT, and sort of what's driving this tumor growth and survival is this increased methylation of H3K27. So EZH2 needs that methyl group donated from SAM in order to sort of serve that function. So if DON is depleting SAM, then you don't have this epigenetic abnormality that's driving these tumors. And so we wanted to see what the consequences of that was in our tumor models. And so we treated with DON, and we looked at histone methylation. You see that decrease of H3K27 methylation. SAM's a universal methyl donor, so it decreases some of the other histone methylation as well. And as a consequence, these tumors are not being maintained in that embryonic stem cell-like state that we talked about was so important to ATRT tumorigenicity. So some of the stem cell factors that you see that are important for maintaining these tumors in that state, SOX2, SOX9, Lin28, are all decreased by DON. And then some of those neuronal differentiation factors that we know are inhibited by EZH2 have increased expression, as you see in C after DON treatment. So we wanted to go back and understand why that was the case. And so we look at DON, which is it sort of inhibits all of the glutamine metabolic pathways, and we see that decrease in the SAM to SA ratio. Similarly, when you grow these cells without glutamine, you get that decrease ratio even more significantly. But the main enzyme in glutamine metabolism is this glutaminase 1 enzyme, and we find that specific inhibitors of glutaminase 1 don't have any effect on this pathway. There's no decrease in SAM to SA. So to figure out what DON's doing to cause this, we did this sort of neat experiment called stable isotope resolved metabolomics, where we can trace how glutamine is donating carbons and nitrogens through these different metabolic pathways, so that we can figure out exactly where DON is inhibiting this pathway. So we treat with DON versus control, DMSO control. We infuse the media with this fully labeled glutamine with carbon and nitrogens that can be traced. We run it back through that mass spec, and we can see exactly how DON inhibits glutamine metabolism. And so what we see, instead of the glutaminase 1 pathway, DON is inhibiting this glutaminase 2 pathway in order to disrupt the SAM to SA ratio. So we know glutaminase 2 pathway is highly upregulated in brain tumors. MYC tends to upregulate this glutaminase 2 pathway further. When we do the stable isotope resolved metabolomics, we get this decrease in glutamate M+5, which is really only could be generated through this glutaminase 2 pathway. And then we see depletion of methionine as well, which suggests that DON's inhibiting this glutamine transaminase K enzyme. And so by doing that, you could sort of see that SAM, as it donates a methyl group, becomes SA, S-adenosyl A homocysteine. And to regenerate SAM, you need that methionine that's being depleted to get back to SAM. Okay. So the next thing we wanted to do is figure out how we could combine it to develop a rational combination therapy to sort of work off these mechanisms of action to improve therapy. And so there's a medication, tazemetostat, that's already in clinical trials in ATRT. It's known as EZH2 inhibitor, but what it's doing is it's blocking the site on EZH2 that's receiving that methyl group from SAM. And so we felt that if we're depleting the amount of methyl groups available for this reaction, then we specifically block where any remaining methyl groups are able to be donated to EZH2. We can more fully inhibit this pathway to really target ATRT. And so we find that in combination, DON combines with tazemetostat, it further decreases H3K27 methylation. And by doing that, it's inhibiting a lot of these stem cell factors as well. So you get this decrease in Lin28, SOX2, SOX9, showing that these factors that are maintaining ATRT in this embryonic stem cell-like state are decreased. We also performed RNA-seq on these tumors, and I just showed you the combination, but we do each medication alone as well. And we find that the combination is change in gene expression, and when you do pathway analysis, many of the pathways that come up are these neuronal differentiation pathways that are upregulated as a consequence of the combination therapy. We then treat our tumor models with the combination. On the left, you see a cell viability assay and formal synergy testing. And we find that in combination, these medications significantly slow tumor growth more than each medication alone. And then we look at apoptosis, which is induced, as you can see, by cleaved PARP. And then our Nextgen assay, too, shows that there's increased cell death from the combination of therapy. So we're anxious to get this into our orthotopic models and see how it affects survival, but we're excited by the results we've seen so far. So we show that glutamine antagonism with DON is a mechanism to target this aggressive mixed subgroup of ATRT. DON itself is impacting the underlying epigenetic abnormalities that drive ATRT. specifically reversing this embryonic stem cell-like state, and it could combine with tazemetostat to further enhance this impact on ATRT. So again, I wanted to thank Children's Cancer Foundation, which really has helped start my career here and helped generate the data that's been important to get additional grants to further expand my research. I want to thank all the people that have been involved with this research. Charles Eberhardt and Eric Robb have been invaluable mentors throughout my young career. Satoshi Nakata and Sabrina Wang helped do a lot of this work in the lab, and our collaborators as well that have been fantastic. So thank you very much, and love to take any questions.
Thanks, Jeff. Any questions?
Wow. Wow. A really good talk, and you had my brain wheels spinning there on all the biochemistry. The methyls were flying all over the place, and my SAM is fully depleted at this point. So I'm not unlikely to get an ATRT today, so that's good. But I want to ask you, so this whole role of EZH2 and inhibiting EZH2 with tazemetostat has been kind of problematic in a lot of other tumors because it hasn't really worked. There's just a lot of clinical trials that were well done that showed an epithelioid sarcoma and others that it just is not going to be effective. So the question I have for you, I mean, it's kind of exciting to see the potential of adding a glutamine depletion component to that to see if that is enough to kick these all over into apoptosis. I'm interested in your thoughts on that. And also, you were saying that the tumors still grow, but they're also apoptotic. So the combination of that, I'm kind of curious to get your thoughts on, whether Don is going to do the trick or whether you think that it's going to be just another help, but not quite solved. What are your thoughts?
No, fantastic question. So tazemetostat, is something that came especially to ATRT where we had the SMARC1 deletion, SMARCB1 deletion. We felt that tazemetostat was going to be the answer. And unfortunately, with so many of these medications, it doesn't end up being that way. This really was generated from Charles Roberts' research many years ago that showed that ATRT become dependent on EZH2 because of the SMARC1B1 deletion. And he found that if you knock out EZH2, all these tumors just melt away, at least in the lab or in mouse models. Tazemetostat blocks that at least some of that methylation activity of EZH2, but never had the results that he got when you just get rid of it completely. The clinical trials have been okay. There's been some responses in ATRT, some really good responses. Better in the sort of extra-renal rhabdoid tumors that have the same genetic abnormality, but not being in the brain means that you get higher concentrations of medication, amongst other things. I think Don's going to help. I think it actually will make a big difference, and there's a couple of reasons why. One is that you're more fully inhibiting this pathway. So one of the problems with tazemetostat might be that you block some of the methylation activity of EZH2, but whereas knocking out the methyltransferase completely, you get these great results. So the problem may be that you're just not getting full blockage of EZH2 activity. So if you decrease the ability for the availability of methyl groups as well as prevent them from binding EZH2, you more fully inhibit this pathway. The other problem is that might be compounded in brain tumors because tazemetostat doesn't get into the brain great, and so you have even poorer knockout of EZH2 function. And one of the things that was exciting about our data, if you look back at the synergy data, where Don and tazemetostat seemed to be the most synergistic was at very low concentrations of tazemetostat, where it was only decreasing cell growth by 10%, 5%, but the combination was still decreasing growth by 50, 60, 70%. So that was the area where it was most highly synergistic. So we're hopeful that because it's so effective at these low concentrations of tazemetostat, where it's sort of working but not great, that might be where it really helps in these clinical trials to more fully inhibit this pathway and bring out the synergy. We show the decreases in cell viability over time, but that may be that it's just killing these cells, and so there's less viable cells left in culture rather than necessarily slowing the growth of the tumors. So I don't know that it's necessarily a counter. I think it is killing these cells and hopefully getting rid of them for good.
Jeff, one question
Yeah.
I have is to what extent do you think the reliance on glutamine metabolism is MYC specific versus, MYC-driven tumors tend to have, higher S phase of cell cycle. You could imagine that having glutamine around is going to be important to any cell that's rapidly growing. So,
Yeah.
to what extent is the effects you're seeing MYC specific, or is it just a general phenomenon for more aggressive, more mitotically active tumors?
Yeah, that's a good question and a good thought. So we find that our aggressive cell models that have low MYC expression, so our BT37 model is one of our fastest growing models, more so than some of the MYC expressing cell models, and it's very insensitive to DON therapy. It takes 50 micromolars in order to reach the IC50. But when we take that model and we force MYC expression, it becomes very sensitive to DON. And, it might grow a little bit faster after the MYC is forced, but, I don't think that makes up for it. It takes 10 micromolars to kill off the BT37 with MYC expression.
The other question I have is there a specific relationship between MYC overexpression and the genomic drivers within ATRTs that would make DON therapy specifically effective in ATRTs? I'm thinking about other MYC-driven tumors like Burkitt's lymphoma, for example. Is it the combination of both MYC activation as well as the underlying genomic drivers that is responsible for it, or could DON be a crossover drug for other MYC-driven cancers?
Yeah, no, very good question. I think a little bit of both. I think so we have used DON in other tumor models, MYC-expressing medioblastoma, neuroblastoma as well, and it's fairly effective. It does something. I think it's a little bit of both, though, because I think this effect on depleting SAM and its effect on EZH2 methyltransferase activity is specific to ATRT, and probably why we see a much better survival effect with DON treatment compared to some of those other tumors.
Great talk. Back to your metabolic pathways.
Yeah.
What would an IDH inhibitor do, or what would IDH mutations do?
That's a great question, and something I don't know exactly what we would see with it. IDH inhibitor would be something that would be interesting to add in. Certainly its effect on hypoxia would be interesting in these tumors, and how it's and differentiation as well. So something that we certainly should look into. Yeah.
Well, great. Thanks very much, Jeff.
All right, thank you all.
Thanks. Sure. Nice try, Jeff.
Almost got out.
Yeah, I thought there was a question coming from the back. I'd seen Pat take off back there. I didn't realize. We've spent a little bit of time looking at the Baf Complex. So for those of you who don't know, the Baf Complex is this chromatin regulatory complex, and one of those is SMARCB1, which is missing in ATRTs. It's also missing in epithelioid sarcomas. And this is where you're looking at as a sweet spot for these kind of combinatorial therapies. And I'm wondering if people have looked at other Baf complex deleted tumors, like breast cancer with some of the units that are missing, and some of these other metabolic pathways, to see if there's, again, this connection between the chromatin remodeling Baf complex and metabolism.
Yeah, no, so I don't know specifically if they've looked at that, but I think something that's especially interesting is this bidirectional interaction between metabolism and epigenetic regulation. It's not something that's intuitive or something that you would think of would necessarily be so intertwined, but these metabolic pathways or the epigenetic reactions are so dependent on the metabolic pathways. These metabolites like SAM that you wouldn't necessarily associate with epigenetics are critical to supporting these epigenetic abnormalities. So, something that's interesting, and I suppose Don and tazemetostat is sort of an example where you combine these metabolic therapies with these epigenetic modifying therapies, you might get additional effect. So it's something that I think is a strategy that's starting to be looked at a little bit more, but I think has a lot of potential for therapies for a lot of these tumors.
Actually, you say it's not obvious, but and it sort of in a way is, because all the post-translational modifications put little groups on proteins, whether it's acetyl, whether it's ubiquitin, whatever it is. It's little peptide, little protein groups, and those all have to come from somewhere.
Yeah.
And your model is elegant because the one transferable small group is methyl.
Yeah.
And we know it's critical for epigenetics. So in a sense, it's not intuitive, but on the other hand, when you get to this point, you're like, oh yeah, of course, it has to be.
Yeah.
So great job, thank you.
Thank you.
Yeah, it's kind of like an epigenetic starvation, right?
Yeah.